Number 6. The first in-human gene editing therapy.
Gene editing has been heralded as a breakthrough technology to treat a range of genetic diseases, and various experimental therapies are now under development. One approach uses a genome editing system, known as CRISPR-Cas9, to make double-stranded cuts in the DNA and repair errors in the genetic code. There are two main methods for delivering gene editing therapies. Remove cells, such as blood cells, from the body, treat them with the therapy, and then add them back. Or infuse the gene editing therapy directly into the patient, either intravenously to reach internal organs for treating a range of conditions, or into the eye for treating vision disorders. This in vivo method uses viruses, lipid nanoparticles, or other molecular delivery vehicles to carry the gene editing therapy to its target destination. One of the first in vivo editing therapies is now in early-stage clinical trials, and, if proven safe and effective, could signify a landmark in the history of biopharmaceuticals. It targets a disease known as transthyretin amyloidosis, ATTR, which arises when a protein, called transthyretin, misfolds and forms abnormal clumps. These protein clumps build up and damage important tissues and organs, like the nerves and heart. The one-time therapy has been shown to reduce the levels of these protein clumps in the blood, a step that could spare precious tissues and halt disease progression. Other in vivo gene editing therapies are also under development and could dramatically improve disease treatment. These include inherited forms of blindness and various genetic conditions that affect the heart, liver, and lungs.